Hello, everyone. Welcome to this new episode of our Student Spotlight series. This is Oliver from CCIR, and today it's our pleasure to have with us Elizabeth Ginsberg from Prior Lake High School. Elizabeth took a course recently with Dr. Strin Murray on computational genetics, and today she's come to share a little bit about her experiences at CCIR and talk a little bit about her projects and her passions. So welcome, Elizabeth.
Yes, thank you for having me.
So Elizabeth, to begin, I think it'd be helpful if you could do a quick introduction, say a little bit about who you are, where you come from, and sort of what your academic interests are.
Yeah, of course. So I am currently a senior at Prior Lake High School, as you said, which is in Minnesota in the US. And at my school, I'm part of a lot of different activities. Currently, I'm the captain of the girls' tennis team, as well as the mock trial team. And I'd say my greatest academic passion is just medicine, which I've always had an interest in as long as I can remember. And I kind of like every aspect of it. I like the research aspect, which is why I took this course. And I also like the clinical aspect. I'm actually part of a program at my high school called the Minnesota Center for Advanced Professional Studies, which is where I'm able to delve deeper into the medical profession specifically and work one-on-one with a mentor, do a bunch of different projects, and I even get my certified nursing assistant's degree at the end of the school year.
Wow. That sounds really exciting. Let's maybe begin by talking a little bit about the course you took and the project that you pursued through the computational genetics course. So I think it'd be helpful first for you to sort of give a quick overview, a non-biologist friendly overview of what computational genetics is as a field, and then maybe talk a little bit about your project, the title of which I'm not even going to try to say because it's quite technical. But yeah, so.
Yeah, so computational genetics, the way that I see it is it's kind of combines coding with biology, in my mind at least. That's how I kind of figured that out. So the genetics part is basically just looking at the DNA, which is the coding for everything in your body and what happens in it. And it's looking at how external factors affect that coding and affect the expression of that. And then also the computational part is where you're able to actually take that DNA and assign kind of numbers to it and then put that into a code in a computer. And you're able to do a bunch of graphs, a bunch of different correlational data and statistical stuff to kind of find out those relations in the body. But you're looking at it at a computer, which I think is really interesting.
Okay, cool. And could you say a little bit now about your project?
Yeah, so for my project, I focused on methylation and acetylation, which I'll kind of get into. But basically within the DNA, because there's so much of it and it has to compress to get into the nucleus, a part of that, histones are a part of that. And so histones are proteins that the DNA binds around that it then can further compress around. And so with these histones, they have a bunch of different tails that help it bind to the DNA. And there are different chemical processes that can change the way that those tails bind to the DNA, either, making that bond stronger or kind of releasing the DNA a little bit more, which can affect how that gene is expressed in the body. And so methylation and acetylation are two different chemical processes that can occur on those histones that then change how that DNA can be expressed. And so I was kind of looking at how those modifications change sense and antisense transcription. So transcription is where the body, it takes that DNA when it's kind of unwound and it connects to it and then reads along the gene in order to express it. So there's usually two strands of the DNA, and the one that the gene is on is usually where that sense transcription occurs, which actually has a gene and will make something happen. Whereas the opposing gene kind of has the opposite coding, And so that's sometimes antisense transcription occurs, which is when the opposite strand is read. However, that strand doesn't actually code for anything in the body. So one of the major questions is, why does the body then choose to read that strand? So I was looking at, how do these chemical modifications that change the way that the DNA is able to be read, and then how does that look at both the coding part of the DNA as well as the we don't know what it does yet part of the DNA.
Okay, so let me see if I understand this. So the first thing to flag is I'm not a biologist, so I don't have the relevant background, but I think maybe in asking this question, I can also help the listeners get clear about this. So it sounds like what you're saying is, so the DNA, as we know, is a double helix. And then there's one sort of half of that helix that is what you're calling the sense part, which
Yes.
is typically the one that is transcribed and does stuff, that gets translated and becomes whatever, bits of the body, or produce certain kinds of proteins, whereas the other part of that helix typically doesn't encode for stuff.
Yeah, so depending on because a gene can be on either one of those strands, but usually, and they can also overlap as well, but usually it's only on one of the strands. So there's actually a starting point and an ending point, and it actually codes for something that will then make another protein. Mm-hmm. So usually the opposite end of that is the exact opposite code. So there really is no starting and ending. Mm-hmm. But for some reason, there is a really high amount of that being transcribed, even though it will die. After it gets transcribed, it doesn't become a protein, so it just kind of disintegrates in the body. But okay, there's too high of an amount of that happening for it to just be a coincidence. Okay.
And the other part of what you looked at is how sort of these two chemicals affect which part of the, which side of the helix gets read, whether it's the sense or the antisense. Is that right?
A little bit.
Okay.
I looked at it really broadly. I was just, are there any correlations? If so, what are they? And then where can I go from there? Mm-hmm. So the first thing I looked at is, how do these chemical changes affect just the normal transcription, just how it's normally read? With, does it, create a stronger bond so nothing can bind to it so it can't be read? Or does it release it a little bit more so then it can be read? And then I was also looking at, well, if it makes a change in the sense transcription, the one that's normally read, is there a change on the other strand as well? So it was just kind of an open-ended question that I was going into.
Okay, cool. And I guess the natural follow-up question is, well, what did you find out? Were there any sort of interesting things that fell out of this?
I'd say so. It definitely changed my understanding of just kind of science as a whole, which I can explain in a bit. But the main thing that we learned during the lectures is that methylations, which is one of the two types of the chemicals I looked at, is usually correlated with lower levels of transcription, so it usually increases the bond, whereas acetylation, which is the other one, usually associates with the higher level of transcription, so it lets that DNA be read more freely. And the main thing that I found in all my coding and graphs and stuff is that that's not necessarily true, that it can be both. Both of those chemicals can have both of those things happen, just depending on which gene it's on. And from this, and then looking at other literature that's already been published, I realized that there's probably such a more complex process happening. It's not really because of this, there's a straight line to this happening. It's probably because of this, then this happens, and then this, and this over here, and then that happens. So it really changed my understanding, because I understand this now a little bit more, but I now have even more questions than I did before, because there wasn't anything that was exactly concrete conclusions, but it just really increased my personal understanding. And another really big thing that I found out is that there's a theory that I found that these modifications, instead of directly influencing antisense transcription actually uses those modifications to then influence sense transcription. So it could be possible that the thing that we don't know what it does, it actually kind of does a reverse and changes how the normal strand is, how often that's expressed.
Cool. So I want to follow up on the suggestive little comment that you made earlier about how this changed your idea of how science works. Could you say a little bit more about that?
Yeah, so one of the reasons that I really liked science like my entire life and why I really liked a biology course that I took in high school is that it seemed a lot more concrete than like other subjects like history. So I like that like you could really like learn it inside out, like how this process happens and how that process happens, and it's this leads to this, and it feels more concrete. But through this project, like I realized that that's really not true. Our understanding makes it seem like it's concrete, and when you learn it in that way. But there's so much that we yet don't know and we just haven't discovered and that we still have to discover, that just those processes are so much more complex, and it makes such an interesting field that you can dive into.
Mm. So I think that's a really interesting observation. Sort of the deeper you get into the field, the more you realize sort of the fact that the stuff that you were learning earlier served more of a sort of pedagogical function rather than say what is actually happening. But so has this sort of, I mean, so you mentioned how, one of the initial attractions of science was that sort of concreteness, but with this sort of sudden realization of the sort of ambiguity of science, or the complexity maybe, does that make you more or less interested in what's happening?
I'd say it certainly makes me more interested in what's happening, because it's allowed me to realize that like there is so much more for me to learn, and I'm like only 16. I'm so young. I have so much ahead of me and in my future, and there's so many different directions that I can go into, so many interests that I have that I can delve deeper into and specialize in my future, which I think is really cool.
Okay, so I want to turn a little bit away from your project and sort of these bigger picture reflections on science, and maybe take a step back and ask you a few questions about sort of the mentorship experience itself, and this experience of doing a research project at the high school level. So one of the things I'm always quite interested in is to ask students how this sort of learning experience, where you're diving into something pretty specific and pretty complex, and pursuing an independent project, how does this differ as a learning experience from, say, your biology course that you really enjoyed in high school? Do you have any thoughts about that?
Yeah, so I'd say I certainly enjoyed the mentorship aspect, especially in like how it was such a small group of students. Like I think there was only 5 in my course, with one professor who was really knowledgeable about the topic. And so I think that really allowed us to, whereas like in high school, it's more of just the course itself, and the teacher only really knows as much as the course allows them to know. So you can't really ask those deeper questions, and you can't really go off topic as much as you might want to with your curiosity. So I think with this program and the way that the mentorship was structured, I was really able to ask any question that I had, even if it was completely derailing from the original topic of conversation. And he would be able to like talk with me about that and answer my questions, and just kind of feed into the curiosity that I had.
Mm-hmm. And what about the writing of the actual research project? Have you done anything sort of at this scale previously? And how does it sort of differ from, say, writing a science or a lab report for the biology class, or writing a paper for your English class?
Yeah, so I've done something kind of similar, not a much like a professional level as this was, but through like I've mentioned a program earlier that I do through my school, and I did do a research paper through that just kind of on my own. That was on like stem cell research. But I'd say that this paper definitely, it differs from a lab report because it's like a combination of the science and English. Like you have to take all this science and all this data, and instead of just saying this is my experiment, this is what I got, you have to explain this is all of the background on it, this is what all of this data means. This is what the bigger purpose is. Here is other literature, you know, that's talked about this. And here's my own conclusion. And here's where I think we should go from there. And I think it differs from just an English paper because it's not just English. It has that science aspect. So yeah, I think it's really just a combination of a bunch of different fields, to be honest.
Yeah. Yeah. And to sort of expand on this combination of a whole bunch of different fields, there's also the coding aspect of your particular course. Could you tell me a little bit about that and your experience with it?
Yeah, so I think the coding aspect was what was most challenging to me. I have had like a little bit of coding experience in like 6th grade, so I'm not an expert, but I think it was both the most interesting and the most challenging. I definitely got stuck a lot because with coding, if you make a tiny little mistake really far back, it just doesn't work. And you have to take the time to find that mistake and correct it. But the satisfaction of when it does actually work is just incredible. So through the program, we used a coding software called MATLAB, which is basically where you input a bunch of different like genetic information, and you can make just kind of a bunch of visualizations of that information through different graphs, which is what I mainly used. And so I was able to, it better let me visualize what I was, you know, talking about and thinking about in my head. And then it also really challenged me in a way that I hadn't been challenged before, which I really appreciated.
So with this kind of data visualization approach to genetics, sort of what, this is just like my curiosity, what do you think that brings to the table that, what does the visualization of data enable you to do as someone who's trying to understand the genome?
I think it allows for an easier understanding, and you're able to see the big picture a lot better than you would be if you just saw the numbers. Because when you're looking at just the numbers, maybe you can only look at then one gene at a time. And whereas you can then, with this data, you can then find like a giant average, and you can create this graph, and you can see like, oh, the big picture throughout the entire genome is this. And you can then connect that to previous experience. So I think it allows you to take this little experiment that you've done and all these really specific data points and create that big picture.
Cool. So I want to now turn from sort of what you've already done to ask a little bit about what you're now interested in doing in the future. So you mentioned earlier that you've had this lifelong passion for medicine. Has this research course given you any insight onto sort of the field of possibilities in terms of your future? Has it sort of in any way informed or shaped how you think of your path moving forward?
Yeah, I'd say it has. I think I really liked doing this course, and I like doing this more research hard science part of medicine. I definitely couldn't see myself doing only this for the rest of my life. So I think what I realized is I love any field that has like a combination of research and then also like clinical like care. So I think my end goal so far has been to become a surgeon, because I think in that you're able to take care of patients, but then you can also do your own research on the side and do things like this course allowed me to. As far as like more of a nearer future, I'm hoping to major in neuroscience because I have a really big interest in the brain. And I think similar to what this course taught me, there's so much that we don't know and so much that I can delve into and find more about. And so, yeah, I think that's kind of how it shaped my future.
Yeah, and I think the emerging interest in the neuroscience is incredibly interesting. Actually in the upcoming session of the Future Scholar, we're offering 3 different neuroscience courses that talk about everything from like brain disorders to brain-computer interfaces. So there's so much interesting work happening in neuroscience, and it's really great that young students are interested in this field. Okay, so I think to begin wrapping up, I want to ask you, well, to wrap up, I want to ask you one last question. sort of more about sort of what you would say to someone who asked you what research was and what sort of the value of doing some project like this would be, especially for a high school student.
Yeah, so I'd say the biggest value of doing something like this that's like a lot more of a complicated level is you really get to feel that independence of doing something on your own and making those decisions and going on a path that might have been completely different from what you originally thought you were going to do. Because a lot of the high school experiences, you're told like, this is what's going to happen next. This is what class you're taking next year. This is what that class content is. This is what your lab is. Whereas something like this, which I think is a lot more similar to what might be happening in college and further on, is you're really able to take that independent track and really you're given that freedom to just follow your interest. And I think that's a really good thing to have.
Cool. That's really great to hear. And definitely for our programs, one of the goals is to create this, what we call like an active learning experience rather than a passive one, where you're really taking charge of what you want to learn and what your interests are and sort of pursuing those projects and interests with obviously the support of someone who's extremely good at what they do and that yeah. But on that note, I just want to say, Elizabeth, thank you so much for your time today. It was so interesting learning about your project and your experience. And congratulations once more on being selected as one of our Spotlight Scholars.
Thank you so much. I really appreciated this opportunity.